Hello everyone, this is Soumya, working as an assistant professor, Department of Zoology, Government First Grade College, Vijayanagara. In this video, I'm going to explain the germ layers. So germ layers are nothing but the group of cells which interact with each other during the embryo formation, and that will contribute for the formation of various organs in the tissues. Except the unicellular and certain animals like the sponges, like which exhibits the protoplasmic and the cellular grade of organization, germ layers can be seen in almost all the animals. And based on the presence of the germ layers, we can categorize the animals into two different types, that is diploblastic condition and the triploblastic condition. Diploblastic condition or the diploblastic animals are the one which are having two dermal layers, that is ectoderm and the endoderm. As the name indicates, ectoderm is an inner layer, and outer layer, and endoderm is an inner layer. In case of the triploblastic animals, the animals which will be having three dermal layers, outer ectoderm, middle mesoderm, and inner endoderm. How exactly the formation of the dermal layers or the germinal layers happens? So after the formation or the fusion of the gametes, eggs and the sperm, there will be the formation of a zygote. After the zygote keeps on dividing into a 2 cell, 4 cell, 8 cell and 32 cell stage division, that forms a structure what we call the blastocyst. Blastocyst will be having the blastomeres in that and the blastocele cavity. The blastocyst, after the movement of certain cell masses within the blastocyst, they'll be keep on the cells of the blastomeres will keep on dividing by absorbing the nutrients which will be provided by the uterus. And the inner cell mass of the blastocyst cyst, they'll start moving, or there will be the movement of the cellular mass, and that is what we call the gastrulation. During the gastrulation process, there will be the formation of the layers, dermal layers, that is the ectoderm, endoderm, and the mesoderm. And ultimately, those layers which form during the gastrulation, they'll keep on continuing the division and ultimately forms the fetus. How exactly we'll see the definition of the diploblastic organisms. The organisms which are having two dermal layers, that is the outer ectoderm and inner endoderm, are the diploblastic animals. Examples are the jellyfish, sea anemones, and various examples can be quoted from phylum Nigeria. Whereas the triploblastic animals which are having three germal layers, that is the outer ectoderm, inner endoderm, and middle mesoderm, are the triploblastic animals. Almost all those the vertebrates, all the vertebrates and the higher invertebrates can be quoted an example for triploblastic animals. How exactly the formation of these endoderm and the ectoderm happens? As I said, as I explained in one of the previous slides, after the formation of the zygote will undergo for the cell division. So in that 2 cell, 4 cell, 8 cell, 30, 16 cell, 32 cell division occurs. After that, there will be the formation of blastula. Blastula, which is consisting of blastomeres and the blastocele. So after the blastomeres provided with the continuous supply of nutrition, so they'll keep on dividing. After the division, there will be the huge number of blastomeres will be accumulated in the blastocele. And those cells which are there in the blastocele, they started moving, or they started pushing the outer cells, the inner mass of cells started moving towards the out, and the outer cells started moving. There'll be a kind of infolding in these cells, and that is the process is the gastrulation. After the gastrulation, there will be the formation of the dermal layers, and of course the inner mass of cells which will get separated and started dividing, uh, so that results with the formation of the ectoderm, and the outer layer of the cells which will be separated from the inner mass of the cells forms as an ectodermal cells, and even that keeps on dividing, forms the ectoderm of the uh, embryo. Whereas there is a structure called the caudal disc, which happens out of the endodermal cells, or the endodermal cells, and that caudal disc which formed out of the endodermal cells, that even start keep dividing and results with the formation of mesoderm. What are the these? This is about the formation of three dermal layers. What are the functions of the dermal layers? So when we look into the ectoderm, as I said, it is the outer layer, outer dermal layer. When it comes to the function of the ectoderm plays an important role in epidermis of the skin, epidermal derivatives like the epidermal gland, hair, nail, etc. And it also helpful in the formation of the nervous system. It also helpful in the formation of the medullary part of adrenal gland, posterior and the intermediate lobes of pituitary gland, and the pineal gland formation. It also forms the eye structures like the conjunctiva, cornea, lens, retina, iris, and the ciliary muscles, and the internal ear, nasal and olfactory epithelia, enamel of teeth, epithelial of foregut and hindgut, some glands like sweat glands, oil glands, mammary glands, salivary glands, and the lacrimal glands. These are all structures are formed out of ectoderm. As I said, ectoderm is an outer layer which form all these structures as I mentioned above. Later, the mesoderm. Mesoderm, as I said, it is a middle part or the that occurs in between the ectoderm and the endoderm. So this will be helpful to form the dermis of skin, the muscle, except the iris and the ciliary muscle. All the other muscle structures will be derived out of mesodermal cells, connective tissues, kidneys, gonads, notochord, heart, blood, lymph vessels, urinary and the reproductive ducts, most of skeleton, coelomic epithelia, pericardium and pleurula, dentine of teeth, cortex of adrenal gland, mesenteries, sclera, choroid of eyes, wall of gut. These are all derived from the mesoderm. So all these, the structures what I mentioned, that will be developed from the mesodermal layer. Later, the last one is the endoderm, which is the innermost layer. So the endodermal structures gives out or endodermal cells. Later on, they will develop into lining of gut, some glands like pancreas, liver, gastric, intestinal gland, thyroid, parathyroid, thymus, and the prostate gland. Larger part of the prostate gland is derived out of endodermal cells. An inner layer of tympanic membrane, lining of the middle ear, trachea, bronchi, lungs, urinary bladder, urethra. All these structures will be most of the vital structures, the innermost vital organ structures will be derived out of endoderm. So this is how the ectoderm, endoderm, and mesoderm ultimately, after forming the three germinal layers, ultimately these layers will divide and differentiate into various structures, like as I mentioned. So, when we look into the germ layers, in case of the diploblastic animals, we can see the derm layers will be the outer ectoderm and inner endoderm. Whereas in case of the diploblastic condition, we can see the outer ectoderm, inner endoderm, and the middle mesodermal layers. So, this is how the cells will be arranged in the diploblastic and triploblastic individuals. To summarize this session, so in this video, I explained the germ layers of various animals. So based on the presence of the germ layers, we can differentiate animals into two different types, that is diploblastic and triploblastic animals. Diploblastic individuals are the one which are having two dermal layers, that is outer ectoderm and inner endoderm. Whereas triploblastic individuals are the one which are having three dermal layers, that is outer ectoderm, inner endoderm, and middle mesoderm. Examples for diploblastic individuals are the coelom trait cnidarians will comes under the diploblastic individual, and apart from them, all the individual except the microscopic protozoans and protistas, all other individuals will come under triploblastic animals. Thank you.